to start. I'll introduce Ryan for coming over. Ryan is a new faculty member at Dana-Farber Cancer Institute at Harvard Medical School. He applies population scale germline genomics methods for topics in cancer disposition and tumor genesis. He completed his postdoc at Dana-Farber with Ellie Van Allen, Kevin Hages, and Sasha Gusev. Recently, and before that, he was a member of Dr. Michael Tarkovsky's lab at MGH for about 10 years, working on GNOMAD and structural variation in germline contexts. He also has a degree in genetics from Dartmouth College and PhD in genomics from Harvard Medical School. Welcome. We're going to keep talking about population genetics and disposition today.
Thanks, everyone. I'm really excited to be here. It's fun to be a whole new crop of people, even though it's just 1 mile down the road. So thanks for having me. It's been great so far. Like Ignati mentioned, I'm a new faculty member at Dana-Farber. Just started my lab last year by way of MGH and Harvard, and then did a postdoc at Dana-Farber. And what I'm going to be talking to you about today is all around sort of the intersection of two fields of population genetics, genomics, and cancer risk, and trying to see what we can learn between these two fields. So the whole scope of our session today is going to be around this notion of why we get cancer. This is obviously a very, very, very broad topic. And of course, there's over 100 years of research into sort of mechanisms of tumor genesis, and just to summarize, a centuries of research in a single pithy slide, we recognize that the major sources of cancer risk are acquired linearly or superlinearly over time, whether it be environmental exposure or somatic mutations. But as a germline geneticist by training, I can't help but be intrigued at the observation that germline genetic risk for cancer is present on the day you're born and remains constant over your entire lifespan, distinguishing it from other sources of cancer risk. Even more so, if you think about proportionally where each category of risk factor is more or less likely to contribute to an observed sort of incident cancer case, earlier onset disease is more likely to have a larger contribution from germline genetics, because there just simply hasn't been enough time for somatic mutations or environmental exposures to drive tumor genesis. So, we'll talk a little bit about early onset cancer today. There's a lot of different definitions out there. Just formally, roughly, we're talking less than 50 years old in an adult solid tumor context. We'll also be talking about pediatric cancers. Net, broad strokes, all histologies, roughly about 5% of patients meet this criteria. Again, hand waving around very important subsets, but just to sort of set the stage. And why, for those of you in the audience interested in translational and clinical applications, should we care about germline genetics in this context? I think really there is a sweet spot here where, if you consider sources of cancer risk, that would be most important to understand if we want to catch patients who are going to develop cancer early, who would be traditionally missed by conventional screening paradigms. Genetics is going to play a larger role in that patient population. Not the only role, but a larger role. And so that's really the translational motivation for all the research I'll be talking about today. Of course, we actually know a lot about cancer genetics too. We have about 100 years of knowledge in this field as well. And again, I'll just try and summarize an entire monolith of scientific research in just a single slide, so excuse me if I'm hand waving through some really critical details, but we'll get to the nuts and bolts later. As of the year, let's say 2026, we now appreciate that predisposition and risk for cancer actually is not so different from other human complex traits and diseases. And what I mean by that is very large studies of common, segregating genetic variation, GWAS or genome-wide association studies, have implicated hundreds, if not thousands, of common SNPs, small variants throughout the genome in cancer risk. Each of these contributes meaninglessly to an individual's patient diagnosis, but net across the whole population, they explain, some meaningful fraction of variation in who does and does not get cancer throughout their life. This can be summarized in the blue bars here. It's for stat gen wonks out there. This is narrow sense heritability, but it's essentially what fraction of cancer liability can we explain due to known common genetic risk factors. And if you compare that to epidemiologic studies, either twin studies or family-based studies from very, very large epidemiologic multi-generational registries, it leads to this observation, which has been around in the field for over 15 years now, of missing heritability or missing genetic risk for cancer and holds true for many, many other traits and diseases. But in many common adult solid tumors, there is a large gap between what we expect the contribution in terms of overall population level liability for getting any of these cancers is and what we can explain with current genetic knowledge. Now, on the other side of the coin, there's a major contribution for individual patients in terms of clinical genetic factors such as rare variants that directly disrupt known cancer predisposition genes. This is perhaps the best known form of genetic predisposition to cancer in the general population. We're talking things like BRCA1, BRCA2, Lynch syndrome families who inherit typically a heterozygous variant in one of these genes that gives rise to a sometimes many-fold increased risk of cancer in one or multiple sites throughout the body. But because these genetic predisposition syndromes and variants are rare, it actually does not explain a very large fraction of all unselected cancer patients. But again, if you tally, so this is estimates taken from the seminal 2018 TCGA study that looked at rare germline pathogenic variants in known predisposition genes across adult solid tumors, 5 to 10%-ish of all unselected adult cancer patients will carry one or more of these rare, presumably pathogenic or high-risk variants. And again, if you compare that to what proportion of cancer patients that self-report a positive family history of that same disease, there's again this gap. So whether you look at the right side common side of the coin or the rare risk factor side of the coin, there's clearly missing risk that for cancer predisposition that we suspect should be genetic. To answer this question, I think we have to revisit the model of genetic variation that we were all introduced to in, maybe in high school, maybe even earlier if you're really advanced, maybe in college. But it's that germline variation or genetic variation in a population is most often comprised or simply comprised of single nucleotide differences between any two individuals' genomes. We've known for over 80 years that this is grossly oversimplifying the complexity of genetic variation that's encoded in all of our inherited DNA. But of course, we do know an awful lot about these single nucleotide changes. I'm showing up here at the top, there's maybe 3-4 million of them in the average human genome. There's also sort of smaller insertions and deletions, 1 to 50 base pair changes throughout the genome. Let's say each of us in this room carries somewhere on the order of 400,000 such variants between any pair of individuals. But I prefer to think of genetic variation in the human population as a continuum of mutational sizes. And so what I mean by that is actually there is a wealth of variation beyond these better studied forms of variation, collectively known as structural variants. They'll be a major theme in the talk today. So abbreviated SVs. Formally, this is any rearrangement of genomic DNA greater than 49 base pairs. That's sort of the definition the field is settled on. And while they are fewer in number than the better studied SNVs or indels throughout the human genome, because they tend to be so large, they actually comprise the single greatest source of human genetic diversity that we know of to date. So they're very, very important, outsized impact on human traits, evolution, and disease, yet for technical reasons that we won't really get into, they have not been studied at scale in many contexts, including cancer predisposition. In my graduate school work, which I have 1 or 2 slides on today just to sort of set the stage for more recent work we've been doing at Dana-Farber, we spent 5-6 years building methods to characterize germline structural variants from whole genome sequencing on the scale of tens and hundreds of thousands of human germline genomes. I was really fortunate to be involved in the genome aggregation database, like Nadi mentioned, or GNOMAD. This is, I think, to my knowledge, still the largest publicly available reference resource for human genetic variation that's out there. I got involved at a very early stage to lead the structural variant characterization in this population of, our initial release was 15,000 human whole genomes. This was it's been published for a long time now, so we've been done with this work for 7-8 years. But I want to highlight one finding, that when we applied structural variant characterization methods to 15,000 roughly unselected humans from the general population, that will bear relevance to all the cancer topics we're going to be talking about today. It is that by going in with genome sequencing resolution, very specialized methods to map out all these different classes and forms of variation in human genomes, we actually realized that germline structural variants comprise most of all genic loss of function events in the average human genome. the upshot here is if you're interested in very profound biologic effects due to heterozygous or homozygous loss of your gene of interest, if you're not looking at structural variants, you're seeing less than half on average of all truncating alleles carried in the human population. What's more is that structural variants, because they are so complex, they can achieve mutational consequences not possible or not accessible to a single nucleotide change or a small insertion or deletion. For example, there's at least 36 or so genic duplication events in the average human genome when assessed across, well, again, we've repeated this, and now we're at 400,000 human genomes that we've analyzed to date, and these estimates roughly hold true, where there's dozens and dozens of non-loss of function, but functionally consequential structural variants that impact protein coding genes throughout the human genome. What does this have to do with cancer? Well, damaging genetic variation in general is important in a variety of traits, but in this initial work in NOMAD, when we look at known, so these are sort of cosmic tier 1 established, no doubt, cancer predisposition genes, and we ask what fraction of people, this is not a cancer patient population, this is just the general population, broadly speaking, what percentage of people carry a rare loss of function structural variant that would impact one or more of these cancer predisposition genes, we come up with a total rate of just under 1%. This is sort of the baseline rate in the general population. If you imagine applying these methods to cancer patient populations, you will expect to see an enrichment beyond this, which means that they're going to explain, we expect, single-digit percentage of all patients, maybe 10% of all patients, so a minority, but a really important minority because these are variants that have very strong effects on known predisposition genes that are being screened for clinically in other settings. And then just one note on sort of, again, the translational arc that I mentioned at the beginning. One of the things that I learned from working on NOMAD is that I'm a PhD scientist. I understand many of us in the room are PhD scientists. For those of us without clinical degrees but want to contribute meaningful impacts to clinical care for actual people out there in the real world, I personally ascribe to this notion that NOMAD showed me and other projects before, that the best way we can do that as a research scientist is to share data openly and widely and make it easy for other people to use. And so I'll illustrate this with NOMAD, is that we spent years and years building this catalog. We released it completely openly 2 years before the paper was published. You could query it online, there was a browser, go look up nomad.broadinstitute.org, it's still there, it's updated with our latest data, you can query your favorite genes, see all the different alleles that we've observed in the human population. It's been really exciting to see this resource grow to date. This is a bit outdated now, but even as of 2024, this resource was being used by 200,000 unique users every week. These are not bots, these are actual humans, clinicians, researchers, people from the public who are just generally interested in genetics are coming to this site. And this curve has continued as we continue to grow this resource. Because it was so widely accessed and because we hammered quality control for years to get the data set to a point where you could trust most of what was being reported there. There are errors, it's not perfect, but we really emphasize the high quality of this data set. Actually, the ACMG, or the American College for Medical Genetics, and the ClinGen Consortium have adopted NOMAD, both for structural variants and for more conventional short SNVs and indels, as one of the pillars of clinical interpretation of findings from diagnostic genetic tests. And to bring it full circle, more recently, there have now been a series of case reports that have pointed to these clinical guidelines from gold standard clinical, authoritative bodies that use the resource we produced on a research basis to find and explain new genetic causes for families with cancer. So in this case, it's a complex structural variant involving MLH1, a known pathogenic gene for Lynch syndrome. That's sort of the full circle as I see it, and that has really informed our strategies moving forward. I'll tell you about today. So what are we going to be talking about today? Well, in the next 30 minutes or so, I want to make sure we save some time for questions. I'll be telling you about a trio of studies that span sort of my postdoctoral phase into some work we've been doing in my new lab at Dana-Farber, all of which sort of are connected by this notion of taking these population genomic genome sequencing strategies, applying them to specific cancer patient populations to identify new and different risk factors for why these people develop disease. In the first, I'll be talking about pediatric cancers. This was an awesome collaboration with also new faculty at Dana-Farber. At the time, we were co-postdocs together with Ellie Van Allen. This was a really fun experience. Then we'll Move on to early onset adult cancer. So this is early onset lung cancer. Very interesting, rare patient population. We'll talk about what germline genetics uncovers there. And then finally, I'll close with one of our more recent studies. Both of these should be pre-printed sometime this spring, we anticipate, where we are digging into cases with unexplained familial cancer. So very extensive family history, no known pathogenic variant, what gives? And I think one of the things that I've learned from these three studies and others, as I've sort of spanned from population genomics into cancer risk and predisposition, is that collaboration is really essential. Finding people who have different complementary expertise than what you have, and both talking about the same problem. Maybe you have different jargon, maybe you have different approaches or understandings. That has been really fruitful. And so just as one example, I joined Ellie's lab as a postdoc in 2022. A year before me, Riaz Galani, who I mentioned on the last slide, who's a pediatric oncologist by training, he sees patients, he does research. He joined as a clinical fellow with Ellie in 2021. Earlier in his fellowship, he had done a very targeted analysis of rare pathogenic germline variants in DNA damage repair genes in pediatric sarcomas. They found some interesting patterns, but it was a very, very restricted analysis based on what would be clinically interpretable at the time. He was interested in structural variants. I was interested in, he had nails, needed a hammer. I had a hammer, needed nails. We got together and we built something kind of fun because in 2023, he reached out over Slack and was like, hey, would you want to chat sometime about, whether we could do structural variant stuff in pediatric cancers? I have all these patient germline genomes. I don't really know what to do with it. Do you want to work together on this? Less than 2 years door to door, we had a paper in print that I'm going to be telling you about today. And so it really moved quickly once we got all the pieces together and found an area where we could both contribute different pieces to the puzzle. In this study on pediatric cancers, our main aim was to understand essentially the natural extension of the introduction I set up, which is we know we can characterize germline structural variants in the general population. What do they contribute for risk for pediatric cancers? For this study, we focused on 3 histologies, so Ewing sarcoma, neuroblastoma, osteosarcoma. One important thing to know is Ewing sarcoma is typically an adolescent cancer. Neuroblastoma, average age of diagnosis is 9 months. So these are, even though they're both pediatric cancers, they're very different etiologically. Net, we had about 1,700 cases. This is all with blood derived, with an exception of a couple saliva, but all with whole blood derived germline genome sequencing, Illumina 30x, 35x, actually. Short read genomes. We had about 1,000 unaffected parents of these kids with pediatric extracranial solid tumors. We had 6,500 unselected population controls that we then matched based on sex and ancestry to the cases, not matching on age for reasons that we can talk about in the Q&A if anyone wants to go into that. And so in total, we had just over 9,000 germline genomes. We applied the tools that I had developed in my PhD to characterize germline structural variants in this population of cancer-free controls and pediatric cancer patients and their parents, finding in total across the entire cohort just south of 300,000 unique structural variants. And one thing I want to emphasize here is that it's easy for your eyes to glaze over at, oh, more genomes, oh, larger sample sizes, oh, the latest, biggest, greatest. Why are we sequencing more people? This is why. Because 84% of the variants we defined in this cohort appeared in less than 1% of all people. As we sequence more and more people, as we analyze more and more people, we will uncover new and different rare alleles that have never been seen before. And one thing geneticists love most is an allelic series. And so this is sort of the case that I would make for analyzing larger and larger data sets, because it will empower the analyses of alleles that have never been observed before. Every patient you add is a movement in the right direction. On average in this study, we got about 7,000 high-quality germline structural variants per patient genome. This did not differ between patients and controls once we accounted for ancestry. We then went on to try and find pockets of these structural variants that were enriched in pediatric cancer cases versus cancer-free controls. The most obvious place to look is rare variants that disrupt genes. So just starting with all 20,000 protein coding genes in the human genome, we computed the rate of these events in cases versus ancestry and sex-matched controls, finding a modest but significant enrichment in gene-disruptive rare structural variants in all three histologies. Osteo drops off, it's very small samples, the data are kind of sparse, but the trend is clear that for all three histologies, these kids are born with slightly more gene-disruptive structural variants in their germline genomes than cancer-free controls. And again, consistent with sort of like a corollary of what we would expect for a true positive, as you filter to rarer and rarer events in the general population, the enrichment gets stronger. Presumably that we are isolating more highly penetrant alleles among this, larger group. But this was all genes. Obviously, not every gene contributes to pediatric cancer predisposition. So we wanted to understand which genes mattered the most, which were explaining this signal we observed. And really fun work integrating gene expression data, both on matched tumors and from normal tissue with the med student Jet Crowdis that got pulled into this project because Riaz and I didn't have transcriptomic expertise, and he was looking to get involved. And again, an example of how collaboration can kind of uncover new cool findings is when we zoom in on the genes that were being disrupted by these rare structural variants in the kids with solid tumors as compared to the controls. We found that in the closest proxy we had available to us for precancer normal solid tissue or normal somatic tissue, those genes that were being disrupted by the structural variants in kids with cancer were more highly expressed on average. It's very noisy, but this is a significant difference in both cases. And then again, when we zoomed in on, so we applied a whole statistical approach that I won't go into for the interest of talking about, the other two studies I mentioned, to isolate pockets of variants that had strongest enrichments in cases versus controls after applying things like gene set membership, expression level, mutational constraint, so on and so forth. One of the striking trends that jumped out, which was really encouraging, is that some of the genes that were most enriched in these loss of function structural variants in pediatric cancer cases versus controls were constrained DNA damage repair genes, which beautifully mirrors a lot of prior work in pediatric sarcomas, as well as Riaz's earlier work that started with a very targeted clinical focus on DNA damage repair genes in sarcomas. But this is a completely different class of variation, and yet it brought us full circle to roughly the same set of genes being important for predisposition to these diseases. We then looked beyond genes, and we turned next to very large germline structural variants. What I mean by this is a germline rearrangement in a constitutional chromosome that knocks out a megabase or more, 1 million bases or more of genomic DNA. These are really, really common in tumor genomes. These are not very common due to strong negative selection in the human population. But the reason we wanted to look here is because based on my prior work in neurodevelopmental genetics during my PhD and earlier years, this class of germline structural variants is the single most frequent genetic cause of abnormal human neurodevelopment. They are incredibly important variants for the development of pediatric developmental disorders, neurocognitive deficits, congenital anomalies. They are included in the gold standard reflex test for unexplained developmental delay in kids. So we reasoned, this class of variation is known to be important in pediatric constitutional congenital disease. Pediatric cancer is early somatic, so it's not a constitutional disease, but it's both pediatric, early onset, severe phenotypes. Maybe there's shared etiology. Indeed, across all three histologies, we found significant enrichments of these very large, rare structural variants in pediatric cancer cases versus ancestry and sex-matched controls. 92% of these, just to get ahead of questions that I imagine you might be wondering about, we also wondered, 92% of these do not touch any gene with a known functional role in cancer of the 700 some odd in cosmic. No cancer predisposition genes touched by over 90% of these. And intriguingly, most of these are actually inherited from an unaffected parent. So they are incompletely penetrant risk factors, but nonetheless have about a 2-to-1 or a 3-to-1 enrichment in cases versus controls. Again, showing that this is a risk factor we should be screening for clinically for predisposition. So we've talked so far about sort of the furthest tip of the age distribution that I alluded to in the introduction. I want to tell you next about another study with a bit less depth that we're still wrapping up right now in a bit less depth. The study has lots of depth. but today we'll only go through a few slides. On the next sort of logical step down this age continuum towards early onset, but canonically adult solid tumors in the case of early onset or young onset lung cancer. This is work that's led by a clinical investigator at Dana-Farber, Jackie Lopiccolo. She recruited a ton of these patients. They're really, really rare and unusual patients, so she really gets the credit for the yeoman's work here. Through a clinical team at Dana-Farber, Jackie and colleagues, and this is a pilot of a much larger expanding study. We're currently working on, several thousand of these patients, but in this initial pilot analysis, Jackie and her colleagues have recruited 251 individuals with lung, mostly adenocarcinoma, lung cancers. There's a few carcinoids, a few squamous in there, but it's mostly adenocarcinoma. That onset before the age of 50. Median age of onset is 37 years old. There's patients that are 16 years old in this cohort who have lung adenocarcinoma. Incredibly rare to see that clinically, terrible disease. All of these patients consented for research and contributed blood for germline whole genome sequencing. We paired that with a published cohort from 2021, so a little outdated at this point. It's the Sherlock Lung Consortium, for those who are familiar, but this is a cohort of lung cancer patients that were recruited for lack of smoking exposure, to understand never smoker cancer, individuals, patients with no personal smoking history. This is more representative of the average age of onset for lung cancer in the general population, 68 years old. So it helps you appreciate just how early onset these patients are here. And then we paired it again in a population way with sex, ancestry, and age-matched adult cancer-free controls drawn from top med cohorts. We again applied sort of not just the methods that I had developed in my PhD, but additional methods to characterize not only structural variants, but germline SNVs and indels genome-wide in these over 2,300 whole genomes. Again, Illumina 30x short-read sequencing. And we did a bunch of analyses. I'll tell you about a couple of these today. So what do we get when we clinically recruit cancer patients at the Farber and colleagues? We do a blood draw. We do Illumina whole genome sequencing, and we throw that data through sort of state-of-the-art computational pipelines. We got 90 million distinct germline variants across a whole slew of mutational classes, a whole slew of mutational sizes, and again, all the allele frequencies, pointing out that the y-axis and x-axis is a log-log, and so again, over 80% of all the variation in this cohort was rare in the general population. This is allele frequency, common on the right, rare on the left. And so just from a single assay, germline whole genome sequencing, which is now available for only $350, I think the Broad does it for these days, we can capture not quite the complete, there's some asterisks there, happy to talk technical details at the end for those who are interested. So there are some gaps, but we can get a pretty comprehensive, pretty complete portrait of germline variation from human genomes at scale. I'm a structural variant guy, so we're going to talk about structural variants first, and then we'll talk about some, moving into other variant classes. But we wanted to understand, again, what germline structural variants do to predispose these individuals, if anything, acknowledging that the null hypothesis is held true until shown otherwise. If structural variants were relevant to lung cancer risk in these individuals with really, really early onset disease. Again, inspired by our pediatrics work, we looked at these large, rare germline structural variants, finding a modest enrichment in early onset cases versus controls. We do not see this in the never smoker cohort, which you might remember is more representative of sort of adult onset lung cancer. So kind of interesting to think about how different risk factors may play into different ages of patient populations, where we don't really see anything when comparing the never smoker patients' median age of 68 to controls. We do see a signal in the patients with average age of 37. Where it gets really interesting, and is a thread that I'll carry forward into the next part of the talk, is one of the nice things about this cohort was that all the patients were recruited through clinicians that were directly involved in the study. We had somatic driver mutational status, driver rearrangement status, histology, clinical follow-up available for over 85% of the cohort. So we, with this data in hand, wanted to know, is there anything unusual about the patients that have these very large germline deletions in their inherited DNA? And we were kind of struck to find that all 100% of the patients with large germline deletions in their germline chromosomes had fusion-driven early onset adenocarcinoma. Sample sizes are small, so I definitely do not want to overinterpret this point. We are continuing to replicate this, like I mentioned, in a cohort of over 1,000 patients, but this holds true no matter which way you slice it. We can't characterize less common fusion partners. ALK is really doing the lion's share of the lifting here. That's just a limitation of the sample size we have. We don't know if it's specific to any one oncogene. We do know that we have not yet observed a lung cancer case that carries a large germline deletion that developed a non-fusion driven adenocarcinoma in the samples we've seen. So hold this thought, definitely some evidence that germline factors can influence somatic trajectories in unique sets of patients. Like I mentioned, in this study, we didn't only look at germline structural variants, though. We also looked at SNVs and indels. They've been better studied, but not as much in the context of really early onset lung cancer, because it's a very rare disease compared to adult onset lung cancer, which is a very important and common, unfortunately common, adult malignancy. And so we reasoned that this might provide an opportunity to discover new predisposition factors for lung cancer. Credit to Noah Fields for actually doing this analysis. He's a master's level technician working with me and Ellie Van Allen. And so we applied an exome-wide approach where we looked for burdens of rare protein coding SNVs and indels in every gene throughout the genome to identify any new risk factors for early onset lung cancer versus controls. Finding TP53, of course. Leaf-Raumaini is not canonically characterized or thought of as a lung cancer predisposition syndrome, although it is out there in the literature. It's more thought of as breast, sarcomas, multi-system cancers that do sometimes occur in the lung. Here we had 8 patients that were germline TP53 loss, heterozygous carriers. All of them were cases. There were no controls. So TP53 in this cohort, in all lung cancer cases, the never smokers and the young lung cases, TP53 was the only gene exceeding exome-wide significance. Again, where things got more interesting is we wanted to sort of stratify this by fusion-driven versus non-fusion-driven cancers motivated by our structural variant analysis. And what we found was that there was actually a gene that popped up at exome-wide significance, IREP2. Don't know much about the gene. If anyone does, would love to chat. But in the fusion-driven subset alone, rare germline variants in this gene, IREP2, are exome-wide significant or Bonferroni significantly associated with fusion-driven lung cancer risk compared to controls. And reciprocally, when we exclude the fusion-driven lung cancer patients and only focus on non-fusion-driven patients, we see not only TP53, but also SMAD6 now pops up as, SMAD6 in and of itself, I'm not aware that is a major cancer driver. SMAD4, SMAD2, other SMADs are. SMAD6 is sort of a redheaded stepsister, whatever, stepbrother of the other SMADs. To my understanding, this, to our knowledge, is, suggestive evidence at a robust statistical significance in a small cohort that rare variants in SMAD6 may be predisposed to early onset lung cancer driven by drivers other than canonical oncogenic fusions. Oh, and then I wanted to point out that one of the reasons even though small sample size, we find it a little convincing is that when we look at where the variants fall, they all kind of 3 of the 4 of the carriers of these variants cluster around the MH1 domain of SMAD6. So functional clustering may be suggestive of there's like a very specific mechanism going on here. We have no idea what it is, but again, we'd love to chat if anyone's interested in that. But if you think about taking the young lung cancer work and what I showed you about the pediatric cancer sort of together, it does lead to this sort of tantalizing notion that perhaps there are inherited germline genetic factors that sensitize people to developing, or their somatic cells, to having unstable genomes and leading to rearranged cancers, whether it be pediatric sarcomas or fusion-driven early onset lung cancers. So what I'm showing here is, this is a perspective piece that came out alongside our pediatric study. It's their words, not mine, but I will repeat it if they say it: inherited genome instability. I don't think we have enough evidence to really conclude that this is what's going on in these cases, but as the saying goes, where there's smoke, there's fire. I think this is a thread that I'm really interested in exploring in larger cohorts and in different histologies, understanding the extent to which the DNA and the variation that you're born with could set you up to be your somatic cells pushed towards less stable somatic genomes versus other more canonical, cancer progression mechanisms. So then, in the last sort of 10 minutes here, I want to talk about another really important axis that I have intentionally left to the side for now. We've talked about really early onset cancer patients. We've talked about early adult cancer patients, but we haven't really talked about patients of all ages who have what looks like a hereditary cancer predisposition syndrome, but have not received a genetic diagnosis. Collectively, these are called unexplained familial cancers. They can arise for a lot of reasons. They can arise through shared environment. They can arise through, genetics as one factor. They can be correlated with socioeconomic factors. So it's not only going to be genetics, but if cancer tracks in a family, if cancer clusters in a family, we know that enriches for genetic cause or genetic predisposition by a factor of about 10x across histologies. So as geneticists, we were really interested to also explore this unusual patient population. These families are pretty rare, and so this is where the population scale strategies we apply came in really big, because there are, as you all are aware, I'm sure, a growing number of publicly available biobanks. Nation-scale biobanks in the US are the biggest one, all of us, but across the pond, UK Biobank, Biobank Japan, there's many such large biobanks that have integrated electronic health record data with germline genome sequencing on hundreds of thousands of individuals. All of us in particular is a data set we used here. We have had great experiences working with all of us. If anyone wants to do similar strategies, happy to chat about how we used all of us. It's a really wonderful resource. We were interested in the genetics, the phenotypes, and the patient self-reported family history surveys on all these individuals, but there's all sorts of other data available in this data set that we did not take advantage of. But what this cohort that now has, I think at last release, it's over 400,000 individuals. This comes from a draw of about 220,000 individuals, V7 for experts out there. What it allowed us to do is take the 34,000 or 35,000 individuals with a positive cancer diagnosis in the All of Us release we were working with, filter down to those individuals who self-reported 3 or more first or second degree relatives with any cancer, not their cancer, important point, keep that in mind, we can talk about that. And then, so that left us with 2,800, 2,900 cancer patients with extensive family history. We then went in and threw out anyone who had a pathogenic or likely pathogenic germline variant from their genome sequencing. That left us with 1,500 individuals who they themselves have cancer. They self-report survey level data. They self-report 3 or more first or second degree relatives with cancer, and they do not have a recognized pathogenic germline variant. We paired that with about 1,200 covariate matched controls. Again, we also screened these individuals for pathogenic germline variants, not that we would expect them to be enriched for it, but just to keep things equal. And we required these individuals to respond to the survey affirming that they had no known cancer history in their first or second degree relatives. So these are like super controls. We've sampled essentially from this biobank from the tails of the distribution. So what did we learn? Well, let's start again with structural variants, and we'll talk about a lot of other stuff that we uncovered. The first thing is that structural variants are a modest contributor to these unexplained familial cancers. Across all histologies, all cases, we found 45 distinct structural variants impacting 33 different cancer predisposition genes, CPGs. So we found things like a 20 kilobase deletion of several exons of MSH2. You could argue maybe this might get picked up in a gene panel test that reports copy number deletions. Not all tests do this. It kind of is a borderline case, but it's also no doubt MSH2 causes Lynch syndrome. This was in a patient with colorectal cancer. We found other potentially more interesting variants in this category, such as a 6 kilobase duplication of a single exon in BRCA2. Based on the splicing, we would expect this to be frame shift. We don't have functional data to confirm that, but also a 6 kilobase single exon duplication is way below the resolution of anything other than like an MPLA assay to capture these exact, very small scale structural variants. And so this illustrates again the value of genome sequencing, which we can get this at scale. We got it in 400,000 people in Nomad, no problem, these types of variants. Some of them will be relevant for diseases and predisposition to cancer. Net across the 18 different histologies, or primary sites, formally I should say. This is using the electronic health record data from all of us. So these are physician-diagnosed cancers. We had to collapse to primary organ. In a lot of cases, we distinguished NETs, neuroendocrine tumors, on the far left. But across all sites, we observed, in general, sample sizes are limited when you are splitting across so many different histologies. But in general, we found that very rare germline structural variants that we would predict to inactivate a recognized cancer predisposition gene were enriched, unsurprisingly, in patients with what appears to be hereditary or familial cancer lacking a conventional pathogenic germline variant like an SNV or an indel. We need more sample sizes. You can see the significance here is barely toeing over nominal. The trends, I think, kind of speak for themselves, and it's also not a super provocative hypothesis. I think this should be sort of ground truth. What could be potentially more provocative is, are there new cancer predisposition genes out there to be discovered? There are people who would argue probably not. And by ascertaining from a very large biobank for very unusual patients and comparing them to ultra controls, we actually are powered to detect different genes at very low population frequencies that would have evaded other studies, sort of population all-comers case control studies. So we again ran a similar strategy to the young lung work, where we tested a variety of sets, formally 5 different criteria for various flavors of damaging germline variants in all of 19,000 autosomal protein coding genes between cases and controls for each of 15 different cancers, using essentially a regression-based approach. SAGE gene plus is the model we're using for those curious. It adjusts for cryptic genetic relatedness, ancestry, all that good stuff. We found in total 5 what seem to be new candidate predisposition genes for adult cancers. One that I think is particularly compelling, this is like probably our favorite, is TSTD2 in thyroid cancers, where it is exome-wide significantly associated with the thyroid patients versus controls in this study. And why I think it's interesting is because, yeah, it's good for an odds ratio of 111, but that's just small sample size. It's an unreliable estimate, but we didn't observe this ever in controls in all of us. It exceeds exome-wide significance, but why we think it's interesting is if you look in large-scale pan-phenome genetic association studies from the UK Biobank as a proxy for replication. For the ascertainment criteria we used, we don't have a direct replication data set. Really, it's not feasible to do that. So we turned to the UK Biobank phenome-wide rare variant association work from Karszewski et al. In Cell Genomics 2022. The strongest phenotypic association with TSTD2 loss of function in the UK Biobank is chemotherapy prescription. The 13th strongest association is thyroid surgery. Why would someone get thyroid surgery if they didn't have something wrong with their thyroid, and if chemotherapy is also strongly associated? This is sort of as close as we can think of coming to replication for this sort of really unique patient population. And then just to give some biological plausibility, if you look at where TSTD2 is expressed in normal adult human tissues, thyroid is among the tissues where it is the most strongly expressed, consistent with inactivation of that gene doing something probably not great in the thyroid itself. We focused so far on on rare variation, but as many of you all are well aware, common variation, and we set it up at the beginning, is a very important contributor to phenotypic variability in populations. This sort of presents an interesting paradox that I would like to show 2 sets of results on in this slide and the next slide, and then we'll wrap up, which I think really boils down to deviating from the Mendelian paradigm for hereditary disease. I'm frankly not a believer in Mendelian dominance anymore, as we've sequenced more and more people. But typically, hereditary disease we think of as a canonical Mendelian model where a single causal variant segregates in a family with who is or is not affected by that phenotype. But what about families that just get a bad draw from the genetic lottery, and every member of that family is drawing from a distribution of common genetic risk that is just way shifted beyond the population average? We tested this in our cohort by taking published, pre-trained polygenic risk score models that essentially sum the common genetic effects of all known cancer-associated, polymorphisms into a single z-score. We adjusted those z-scores for ancestry and other covariates, and in almost every cancer we looked at, we found the case distribution in the color was right-shifted, indicating an increased enrichment for polygenic risk, common genetic risk for cancer compared to controls shown in gray. I think almost all of these are at least nominally significant, but one thing to note here is that not all polygenic risk scores are equally performant. Breast and prostate have some of the best polygenic risk scores of any human disease out there, and you can see that those p-values are not ambiguous. It is a statistically significant shift in increased risk in patients with unexplained hereditary breast or prostate cancer, for instance, compared to controls. And why we believe this actually has some relevance is because we cross-analyze these results with family history. And so in thyroid, for instance, here's the control distribution. If you look at cases every case in this cohort had family history of cancer, not necessarily the same cancer they were diagnosed with. If you look at the thyroid cancer cases who had cancer family history of something other than thyroid cancer, they're not that different. They're statistically not different from controls. Maybe there's a slight shift favoring increased risk, but not statistically significant at this sample size. If you match on family history, you then see this, it's a modest shift, but it is a shift favoring more polygenic common risk concentrated in thyroid cancer patients who have family history specifically of thyroid cancer. That makes sense, right? We can look the other way. So let's look at colon, control distribution for colorectal cancer. And what we're going to do here is we're going to first look at patients who they themselves did not have colon cancer, but they have another type of cancer, and they reported a family history of colon cancer. So they might have ovarian cancer, and their mom might have had colon cancer, something like that. This is a significant difference in the cases have slightly more risk. We're better powered here. We're talking, this group is 320 cases, so we're better powered to find smaller effects. It's not a huge shift compared to when you match family history. Yet again, you see this group pop up even more. And again, polygenic risk, it's a weak effect, but it's quantifiable in every single person on Earth, which means that this generalizes strong genetic effects when summed from all sorts of common variation to a test that could be, run in anyone for determining, inherited cancer risk beyond just, these Mendelian predisposition syndromes. And then, so the previous slide talked about what we can learn when we aggregate genetic effects across the entire genome from common variation. But another way we could think about common variants is a way to look for blocks of the genome where just by chance individuals have inherited identical copies of DNA, so runs of homozygosity. You could imagine that this might unmask maybe recessive loci that haven't been picked up by large-scale studies where typically you assume an additive model. And so we tried this, and it worked in a handful of cases, all of which were actually really convincing, but this is not, it's coming at the end to sort of as an interesting tidbit, not like a big explosion, where in kidney cancer, for instance, we found a genome-wide significant locus, or a locus on chromosome 22 down here that had a genome-wide significant excess of homozygosity in thyroid or kidney cancer cases compared to ancestry sex-matched and age-matched controls. When we look at this locus, here's what the actual data look like. The green is sort of a stepwise average of the fraction of patients that are homozygous at this locus. Gray is controls. You can see there is a really striking increase in homozygosity over this entire region due to the haplotype structure of that region. This is sort of why common variants are valuable. But there's a ton of genes here, and so we were left with sort of a fine mapping problem, like are any of these genes likely causal? What's going on here? What's the mechanism? Well, again, by comparing to kidney cancer association statistics due to rare variation in the UK Biobank, a separate data set, one gene popped out, BPIFC in particular, as being provisionally associated with rare damaging variation in kidney cancer in the UK Biobank. So to make a long story short, we don't know the mechanism here, but it is more than a coincidence that the unexplained kidney cancer cases, familial cases in this cohort, have such an excess of homozygosity over a gene that has a provisional association with rare variation in the exact same cancer type in a different data set. More work obviously needs to go into this to understand what's going on. So statistically, if we try and summarize everything that I just threw at you for the unexplained familial cancer portion of this talk, population attributable risk model, for those familiar with it. It's essentially trying to regress how much of case control status can be explained by the sum of all risk factors that we've looked at here. And so it'll result in a value from 0 to 100% of case control variance, essentially explained by these factors, controlling for ancestry, sex, other covariates. And we find, we don't explain all unexplained familial cancers, but a few trends jump out here, which is that we explain up to 18 by looking at known loci. So these are structural variants in genes that we already know are involved in those cancers. This is polygenic risk already established to be a risk factor for those cancers. We find in the case of prostate up to 18% risk is explained in these cases just by looking at risk factors that we already know about. And then if you add in, and this is where it gets a little sketchy with Winner's curse, that happy to chat about the confounds there, if you add in the new loci that we discovered, like the TSTD2 thyroid cancer predisposition candidate that we discovered, we actually add 2.4 to 20.5% additional variance explained here. So we're not explaining more than half of all unexplained familial cancers with genome sequencing, but we're explaining double-digit percentages by applying genome sequencing and looking at loci we know, as well as trying to find new loci and new genes that have not previously been implicated. So I'll leave you with 3 takeaways here. The first is that I'm not trying to sell this, but I do sincerely believe and have hitched the long-term success of my career onto this notion that germline genome sequencing is ready for prime-time applications in cancer genetics. And by using these techniques, we can uncover genetic mechanisms of cancer predisposition that have evaded, for a variety of reasons, technical and otherwise, prior studies in cancer risk and tumorigenesis. And the thread that I highlighted sort of 2/3 of the way through the talk that I'm really excited to continue thinking about and would love to collaborate in this space is to what extent germline genetics predisposes individuals to following different somatic evolutionary trajectories in the evolution of their tumors. I think this is a really interesting area that bears deeper exploration. Like I mentioned, I just started my lab at Dana-Farber last year. We are, unsurprisingly, continuing to scale these approaches across. We've now done 60,000 germline whole genomes for 17 different solid tumors. That work is still ongoing and a little early to talk about today, but maybe in the future we could chat about that. Like I mentioned, another big area of interest is understanding how do we get from healthy tissue to cancer and what role genetics plays in that. Minor role, but maybe an interesting role. And then ultimately, bringing it back to the beginning, I would love to have a translational impact from all of this work where we can figure out who is at highest risk for early onset cancer so we can screen them earlier to save lives. I'd like to thank my 3 mentors for my postdoc, everyone who's been involved, and happy to take questions. Thank you.
Fantastic talk. Any questions?
All right, I'll start with asking, in all this sequencing of germline variants, how often do you have also matched transcriptome data to actually assign a meaningful change in the transcriptome that's associated with the cancer phenotype of the sequencing?
That's an awesome question. We are working on hiring someone to answer that because we're sitting on a ton of data and we just haven't analyzed it yet. So we have in our adult solid cancer cohorts, we have close to 6,000 blood-derived bulk RNA sequencing data sets, and we have close to 5,000 matched tumor short-read transcriptomes. So net, it's over 11,000 total bulk RNA sequencing data sets that are matched into patients for whom we've analyzed their germline genomes. It's a mix of tumor normal, and the overlap between the tumor and the normal RNA-seq is not 100%. So that work is very primordial, but I think that's a very, very logical next step of linking functional consequences here. We have looked at that in previous studies outside the context of cancer. So in corollary work to NOMAD, we've looked at the functional consequence of structural variants, finding more or less roughly what you would expect. Deletions of any coding sequence, germline deletions, lead to roughly a 50 or 40-ish% decrease in average expression level of that gene. Gene duplications lead to a 50% increase on average of the corresponding transcript that's being duplicated. Other types of structural variants are more complicated and have multifarious transcriptional consequences, but I think that is definitely an area that we want to go to next, try and link it to actual mechanism. And it'll require the data sets exactly like you mentioned.
I have a question about germline inheritance.
Yeah.
How often are those your structural variants?
Yeah.
How often are they de novo, such that they are not in parents?
Yeah. So in the pediatric context, tell that question's coming because there's data. So we expect. This is work from Nomad using the Watterson estimator, which has its limitations, but is very useful for population genetic studies, to suggest that we think the true rate of de novo structural variants occurring in live births that we can capture with short-read genome sequencing is about only one new de novo structural variant every 2 to 6 live births. That's a rough range. So most people do not have one. When we look at Ewing's or neuroblastoma, and these numbers are restricted to the complete trios that passed all of our filtering, so we manually reviewed every single one of these candidate de novos. There's only one that we flagged as false positive. Everything here passed clear manual review for a true de novo structural variant. We see that the rates here are all not really significantly different. They're maybe in neuroblastoma, maybe it's slightly elevated, really small sample size, only 100 complete trios. We found 24 de novos out of 103 complete trios we were able to assess. Maybe that's slightly elevated, so maybe there's an elevated rate of germline de novo mutation in the early onset disease, but it's not a whopping difference. So I think really what it boils down to is of the de novo variants, when they do occur, they are just larger and worse. They impact more genes. So here's size being shown for the de novo events compared to polymorphic variation present in one or more individuals but inherited. And then this is the proportion that damage direct coding sequence in dark green. De novo is a whopping enrichment there. And so I think when these do occur, in kids who develop early onset phenotypes, they're going to hit parts of the genome that should not be tampered with. I think that's the best we can answer.
For those large structural variants, do you notice anything like at the break ends? Is there anything interesting about the sequencing of the break ends?
Yeah, that's, as you probably know, that's been characterized really well in these recurrent genomic disorders in peds. We actually see that they just seem to pop up sporadically throughout the genome. That said, we haven't done, like a microhomology analysis, but there wasn't an obvious overt enrichment at specific, like NAHR mediated events. I think I have a slide in here. Yeah, so this is what some of them look like in terms of their copy number profiles. They're huge. They adhere to strict integer copy number states, as you'd expect for a true germline event. And when we annotate these breakpoints with genomic features, there's nothing really obvious. Sample sizes are really small, so maybe it's the case that there's a trend, and maybe we haven't looked at the right thing, but it definitely is different from what we see in peds, which is large tracts of segmental duplications, non-allelic homologous recombination, big deletion or duplication. It's a different mechanism here, we think.
Have you seen any sort of association with somatic SV signatures for any of these?
We are really interested in that. We looked when you index in on patients that have matched somatic sequencing in the peds context, we didn't have enough to say. It was so noisy. We didn't really have enough sample size to concretely say too much outside of anecdotes. Let me see if I have. I don't. So there were isolated cases where we found an individual with a confirmed de novo duplication of a few hundred kilobases of MYC-N, which is a known oncogenic driver of neuroblastoma. This is a clean 3-copy duplication. We ruled out tumor and normal by applying DT and other methods. And that patient's tumor shows like a 50-copy amplification of MYC-N. So it stands to reason that there probably is some, whether it's early post-zygotic mosaic and we're calling it germline, we can't rule out, but the fact remains that that tumor is addicted to MYC-N at a very high level, and that patient's early developmental, whether true germline or early post-zygotic mosaic, included a constitutional duplication of MYC-N. So that's like an N of 1 example. I think adult cancers will have much more data to answer that. These peds cases, the data was really ugly and not large sample sizes.
That was actually my follow-up question. Have you seen any mosaic behavior for any non-integer behavior. Right?
Yeah, so 99.9% of all variants we call when we look in trios are clearly inherited. Of the 0.1% of all variants that appear de novo, almost all of them are false positives. A small handful of them are true de novos. So of the ones that we consider true de novos, 7% of those appear either mosaic in the parent or mosaic in the child. So not complete integer copy state in the child, but so presumably de novo in the parent's germline, or no, excuse me. You know what I mean? Either mosaic in the child or mosaic in the parent. Yeah, so it's a small fraction. I think we're very strict in our filtering, so I think the true fraction, if you were to drive down your detection threshold, accepting more false positives or maybe fancier methods, there's probably more. There's certainly more to be captured. Our estimate is of the de novos, we find 7%-ish are putative mosaics.
Great. Thank you so much again.
Yeah, we have for a subset. Since the data are collected, so that's why trio, our samples are but we have larger samples for the osteo.
Right. And those osteos has characterized these people do
it's totally off my radar.
Because that would be super
I know that
parents, right?
it was brought up by
Because that's something that nobody's doing. That would be super interesting.
Yeah, so we have
leadership.
I'll need to check on the trios versus
Yeah.
for osteo.
Yeah.
It's about 50-50 for neuroblastoma and humans are less interesting.
Well, neuroblastoma might be as well.
Yeah.
Because there's a high levels there as well.
I forgot.
Is someone going to get
Yeah.
Ryan
Definitely not
after
you.
Mark
So the neuroblastoma we have. I mean,